First of all, I will talk, my speech is out from the toxicological point of view. So I will continue. Just an introductory picture about National Food Agency. We work for safe food and drinking water. This is my agenda, a little about state of the art. I will also touch on the morning session a little and suggest the ways ahead and outlook for the future with sustainability and so on. And also a little about our tool, the Risk Thermometer. Today we are faced with different problematic aspects regarding substances. You have oligomers, NIAS, nano, EDC, and antimicrobials, and also many different kind of materials. And challenge for agencies, for example, Food Agency, NFA, there are so many substances used in packagings. Just a few are evaluated every year by EFSA in plastics, just a few. And we found even less amounts. We have better analytical chemistry methods. And as a toxicologist, we discuss a lot of low-dose effects, endocrine disrupting chemicals, and mixtures, cocktails, with other words. And we have environmental requirements as well, and also economical problems, less resources in money and toxicologists and so on, and other priorities. This slide just shows that in 2011, the EFSA Working Group in cooperation, EFSA, found out that in national regulation within Europe, there are all these in red substances were non-evaluated, and the ones in green were evaluated. So there are many non-evaluated substances, at least according to the EFSA guidelines. And some other publications showing the same, for example, in printed paper and board, you found a lot of substances which are not evaluated. And also there are substances which are evaluated and authorized, but still of concern. So how do the National Food Administration work, agency work? We have small resources, so therefore we are thinking more of what are we exposed to. We're using biomonitoring. And with biomonitoring, you could find trends, and you find the real total exposure, not just from food contact, from the food itself, from dermal exposure, and so on, other sources. And we also try to use a little dichotomics. We have been running a project since 1996, which is called the Pop-Up Project, Persistent Organic Pollutants in Primiparous Women from Uppsala. And the goal is to find out trends about chemical exposure. And we use that by sampling of mother's milk, hair, urine, and so on. And here you can see what has been monitored over the years. Some of these substances are very common, of course, in food packaging. That's why it's very interesting to speak about this. We have the perfluorinated alkyl acids, phthalates, bisphenol A, triclosan, etc. And here you can see the trends, for example. This is in urine, DINCH, an upcoming phthalate, which have been replaced the DHP. And for BPA, it's the downward trend. And then we have BPF, bisphenol F, it's an upward trend. We have also collaboration with the group working on dichotomics. That means that you try to identify unknown substances using human samples, human hemoglobin or human DNA, for example. And then you use techniques like LCM, SMS to find out about these adducts, it means a product. And we combine that also with the in vitro gene tox testing, for example, the micronucleus test. We take the same blood sample, for example, for the same person. But what about safety then, when we think of the future? Isn't there a contradiction between environmental criteria and food safety? Here you can see we have bio-based material. Is there any problem with that? Do you have natural toxic compounds for reuse, for recycling and so on? What about the many unknowns and so on? So there might be contradiction. So what I now would talk about is some suggested ways ahead, especially on evaluation in that respect. So I would like to have more data sharing between, for example, EFSA and ECHA, because even when they are thinking of toxicological dossiers and so on. And when we are thinking of how to find out unknowns, we are talking about mixtures now, maybe when we have a recycled material. A very promising way has been, published from the Danish authority, where they have tried to do some kind of top-down approach. They have screening packagings, and then they, in combination with a bottom-up procedure, where they know the chemical and they test it in bioassays, so they know the chemical. They combine that with the top-down approach, where they screen packagings. Here, yeah. So we have to make use of more in vitro bioassays, but we also have to know what we are sometimes we have to look for. You have toxicology, you have biosafe, and so on. Sometimes you have to combine that also with computational toxicology in silico methods. And after that, you may even have to do some more real tox studies, in vivo studies. But at our agency, we also think that you should be very interested. What's getting into my body? That's what are we exposed to? You can measure substances in packaging, and so on, but still you want to know what gets into my body. What am I exposed to? So therefore, we think that biomonitoring is one of the clues for the future. It's already been done for many years. And also exposure should, when we relate substances, they should be exposure-driven. EFSA has just turned over to exposure from migration. Just a few words of some further suggestions. Transparency and better risk communication from agencies to the consumer, for example. Collaboration. In the Nordic countries, we used to have a lot of projects, and also the Council of Europe was more work before. Now it's less, I would say. And instead, we have the EFSA network. You have EU reference laboratories and so on, and we have joint projects. And also, of course, mutual recognition, if it's possible, harmonization. Traceability, what's in the source for recycled materials, for example. And maybe we should develop the rest also for more information, just not for informing about exceeding limits and so on. This shows just some Nordic collaboration progress over the years. Some of these Council of Europe cooperation progress which ended in resolutions. I just want to say a few words about the Risk Thermometer now. It's for risk communication, especially to the consumer, media, other stakeholders. It's also used for priority setting, what substances would we focus on at our agency. This is the traditional risk assessment. And you have a tolerable daily intake, which you shouldn't exceed, for example. We have developed something like a severity-adjusted marginal exposure approach. And then we have defined that into 5 risk classes. So instead of just looking at the effect, we look also at the severity of the effect. Here it shows that you have markers for increasing severity. And for example, some of them are in liver, for example. So depending on publications and what they have produced, we have used this severity to put into our model or tool. So this slide shows that, for example, I have difficult to see here, but you may have lead, for example, in risk class 3, you see the gray part, and in risk class 3, you have a dioxin in risk class 3, you have cadmium in another risk class, and then you have hexachlorobenzene, and at the end, the bisphenol A. I mean, first of all, you need a good exposure assessment. We had used the market basket surveys, and we have also other food surveys to look at the exposure. Then you compare with a reference point from an animal experiment. And then we have also added this severity factor. So for the average adult in Sweden, you can see that bisphenol A, no concern at that risk. And then up there, that's for the moment, of course. Okay. So you will see that when you get into the red, it's more, you have to take some action. So this will use for the consumer, for example, now. So it's better way of more precise risk communication when media goes out with some media scare. So conclude, especially about evaluation, more data sharing, screening of materials in combination with bioassets, and using more of human biological samples. Prioritize what substances are important for you from the food agency point of view, food health, and so on. And then, of course, transparency, collaboration, legislation, you can do more. Thank you.